The overall goal of this procedure is to demonstrate a quantitative DNA binding assay, which is useful in examining protein DNA interactions, discovering transcriptional regulators of gene expression, and in identifying novel anti-tumor agents. This is accomplished by first preparing a streptavidin-coated assay plate with an appropriate biotinylated DNA target. The second step is to add nuclear extract proteins with or without potential inhibitors and allow them to bind with the DNA target. Next, the bound transcription factors are detected with primary antibodies and horseradish peroxidase-coupled secondary antibodies. The final step is to add a horseradish peroxidase substrate to the wells in order to elicit color development. Ultimately, color development is measured with a spectrophotometer over time. The amount of color change will indicate the degree of DNA binding and the effect of potential inhibitors of DNA binding.
The main advantage of this technique over other techniques such as electrophoretic mobility shift assay is that it is a quantitative DNA inhibitor binding assay.
To begin, prepare an adequate number of cell cultures consisting of at least 3 × 10⁷ cells for each condition tested. Here, human bone